The demonstration of gas chromatography. How gas chromatography is working, because we are using two phases. One is the stationary phase and the other one is the mobile phase. The stationary phase is a non-volatile liquid we are using with some supporting material, whereas the mobile phase is basically the carrier gas. Basically the carrier gas most of the time we are using is hydrogen, which is having the better thermal conductivity. So we are using hydrogen as the carrier gas, right? So here we have three cylinders here. The first cylinder is basically the hydrogen gas cylinder, second one is zero air cylinder, and the third one is nitrogen gas. The nitrogen gas is specifically used here for flame ionization detector. That is the reason we are using the nitrogen gas as the third cylinder. And if you look at these cylinders, in all the cylinders we have two knobs. The upper knob is 0 to 15 pressure and the lower knob is 0 to 250 pressure, right? So what we have to do is, we have two options here. One is open, another one is close. With the help of the driver, we can either open it or we can close it. Generally, when you want to perform the gas chromatography, first of all, you have to open these cylinder walls. When we are opening the cylinder walls, we have to adjust that within 5 to 6. It should not be more than 8 kg. At any cost, it should not be more than 8 kg. If you see here, it is also around near to 5 kg, right? And if you see this also pressure, it will be near to 5 kg, right? At any cost, it should not be go more than 8 kg. It will be very, very dangerous. So we have to maintain the pressure of the cylinder PSA when we are opening. It should be within 5 to 6 kg at the most, right? So after opening these three cylinders, now this is connected over there, right? So there we are having the regulator here. The pressure regulator and the flow regulators will be there in this area. So if you look at this pressure regulator here the gas control system is present, right? Here we have the moisture absorbent will be there, right? But if any moisture is there, that will be removed as such. And here it is a hydrocarbon. Here there is a hydrocarbon. Here there is an oxy trap will be there, right? And this thing basically, if you look at here, this is basically for the hydrogen gas. The second one, blue, is basically for the zero air, and the yellow is basically for the nitrogen gas. And when we see here, there also the pressure, if you look at that, almost the needle will be looking in a similar manner. That means at any time, we have to regulate the pressure here. At any time, it should not go more than 5 to 6 kg here, right? And here the another important thing is that here the bottom, when we see the bottom, because this pressure, whatever we are keeping here, this only will be going into the column. So therefore, what we have to do, this pressure, there is a knob here. You can take any knob. Here there is a knob here. For example, if you look at this knob as such, I can just take this knob here. If I look at this knob and then I can rotate here. So what will happen? This will be moving here. Can you able to see? After rotating this thing, then I can just press it up. That's all. Now it is off. The valve will be off. If you want to just open the valve, what we have to do that, you just click it like this. It will be opened. Now the gas will be opening and that will be flowing inside the column, right? So this is the way we can just open the valve also, right? This is the shut position of the valve, and for opening we have to just press it down, right? So automatically it will be opened and the gases, all the three gases will be coming inside the column as, right? And in the column we have two important things. One is called auto injector, AOC 20i. We will be having the auto injector here. So this auto injector, basically what we will be having, if you look at here, this auto injector, if you look at here, this is a sample compartment. There is a sample compartment here. So whatever the sample compartment, there is an auto injector here. You can able to see here, there is an auto injector here. So inside this, automatically they will inject the sample. But before that, we have to just put the sample and we have to dissolve that in a suitable solvent. The sample, whatever we are using, may be either volatile or semi-volatile samples. So these samples, we have to put it in the tray and then we have to keep it over here. Yeah, 1 minute. Yeah. It's taking time a little bit. Yeah, now you can see. So this is the way we can attach this knob, and after that this is your column. The column actually we are having, now I have not opened the valve. When we open the valve, automatically the gases will be flowing in, then automatically the temperature will be increased inside the column. So all these things we can do with the help of the software. So here the name of the instrument is Shimachu GC2010 Plus. This is the name of the instrument, and here all the knobs will be there. Automatic and manual knob is also there. This is the manual panel. We can do manually also. If we want to start, we can start it, and if we want to stop, we can do the stop button as such, right? And here inside this column, if you look at this column, this is an auto injector we are using, AOC auto injector, right? So in this auto injector, the tray we have to prepare the sample and keep it. Automatically when you switch it on, the tray will be kept inside and it will do the automatic injection here. And this is our column as such. If you look at this column, this is a column we are having as such. The column is basically, if you look at this column is basically made up of polydimethyl siloxane, right? So rested column, because you see the diameter of the column is very less, 0.25 millimeter diameter, because there are different columns we are using. One is called packed columns, one is called open tubular capillary column, another one is called wall coated open tubular columns. We have the packed columns, then we have WCOT columns, that is the wall coated open tubular column. Another one is called SC coated columns, that is the support coated open tubular columns. So there are different types of columns we are using. Preferably the open capillary columns are better, because these capillary columns, the sample size is also very less, and automatically we can use all these things in a proper manner. That is why, and remember when we talk about the injection, there are different types of injection. One is called the split injection, another one is called the splitless injection, and the third one is called on column injection. So basically we are having the split injection. We can use the split injection as well as splitless injection also. So most of the time, depending upon the sample, if your sample is very less, you can go for splitless injection. If your sample content is little more, then we can go for split injection, which is more advantageous also, right? And inside the column, if you look at here, this is the temperature of the column will be maintained. See, now I have not switched on the things. Once I made the things on, automatically we cannot open in this manner, right? So the column basically is made up of a different type of columns. The stationary, it is having the stationary phase coated columns, right? And you can see the minute tube is there, right? This is the capillary tube, minute open capillary tube will be there, which is coated inside the column as such, right? Now, when we talk about these columns, basically when we talk about these columns, we have two important aspects. The column, either we can use the polar stationary phase, or sometimes we can use the non-polar stationary phase. Polar stationary phase column is basically carbovax or polyethylene glycol, whereas non-polar stationary phase is basically polydimethyl siloxane, and intermediate polar also we can use the polydimethyl siloxane as a substance inside the column, right? And the column temperature, ideally we can maintain the temperature up to 350 degrees centigrade, right? 350 degrees centigrade, not more than 350 degrees centigrade we can go, depending upon the sample. Some samples we can go for 250, some samples we can go for 300 degrees centigrade. Maximum temperature tolerable by the column is 350 degrees centigrade, right? So this is what we have to do automatically from the column. Now here, if you look at this in the column, you can see here there is a tray over here. Can you able to see here? I can show here this one. This one, there is a tray here. So this particular area, so if you look at here, this particular area, this is basically the detector. The detector is above the column here. This is basically the detector. The name of the detector is called flame ionization detector. Of course, there are different detectors we are using, but that is the reason. The detector also has to operate properly. That is why we are passing the nitrogen gas. Right? We are having the zero air cylinder, then we have the hydrogen gas. Hydrogen gas is the mobile phase, which will take your sample along with that. Sample will be passed here. We put the sample, then the mobile phase will be coming in, and the mobile phase will be saturating your sample, and then they will be taking inside the column. And from the column, the sample gets separated, and they will go into the detector. Detector is here, right? As I already told you, this detector is basically flame ionization detector, where any hydrocarbon sample which is present in the sample, they will be catching the flame. They will be ignited and they will be producing the cation, CHO plus cation. Right? When carbon and hydrogen will be there, definitely they will make the CHO plus cation. So this cation, whatever produced, they will be traveling towards the tip of the flame, that is your anode, and from that you will get the signal response, and that signal response will be detected by the detector. So flame ionization detector is the universal detector. It is the detector mostly used for most of the samples. It is the common detector we are using. Universal detector is another one that is called the thermal conductivity detector, also called the cathometer. So specificity will be less with universal detector, thermal conductivity detector. Therefore, here we have procured the flame ionization detector, which is the most widely used detector, and most of the samples for gas chromatography which contains the hydrocarbon can be analyzed by this flame ionization detector. But there are certain other detectors are also available. One is called electron capture detector, which is especially meant for using or capturing the negative compounds like halogenated compounds, insecticides like parathion, malathion, all these halogenated compounds can be trapped with the help of electron capture detector, right? So this is all about the detectors, and this is about the instrumentation. There is one software called Lab Solution software. Using this software only we are performing the gas liquid chromatography. We have the instrument here. If I click the instrument, there is a post run, administration, manual, lot of icons are there in the system, and particularly in the desktop. If you click the desktop, right, it is just opening. So you can control it manually here, the column temperature, all these things you can control it manually, or else you can also do it with the help of the software also. So software, you can see system configuration will be there, system check you can do, data acquisition, real-time batch, and then report format. Everything you can get it here. So this is an icon we are using, right? So finally you will get the report format also here, right? So this is all about the instrumentation, particularly in file, all these things, instrument system configuration. If you want to switch on the instrument, you have to go for system configuration, right? So all these things, monitor will be there, tools also will be there, windows, all these things are present here, right? And here you have to adjust this thing. Now I have not connected so far, that's why it is showing not connected. So whatever the temperature, once I make this valve on, automatically then it will be connected. After I do the programming here, it will get connected, right? Then I can keep the temperature here. What is the temperature, flow, purge flow, column pressure, column temperature, and FID temperature. FID temperature is also important. Detector temperature is also important. The makeup flow is basically the nitrogen gas. And FID hydrogen flow, air flow, all the things will be identified here, right? So this is how we are using this gas chromatography by Simatsu as such. I hope it is understood for you. Thank you, my dear students. Thank you very much.